Hello everybody, I'm Dr. Dayashankar Gautam, Assistant Professor, Department of Zoology, St. Aloysius College, Jabalpur, Madhya Pradesh. I'll be preparing a video series on human genetical diseases. This is the first part in which we are studying Down syndrome. Now, what are syndromes? Syndromes are a group of characteristics which are expressed in the phenotype of any organisms, and these syndromes are caused because of structural and numerical changes in chromosomes. Now, to know the karyotype, what is karyotype? It is a picture of complete set of metaphase chromosomes which can be studied for chromosomal abnormalities. Now the types. There are two types: autosomal syndrome and sex chromosomal syndrome. Now what is autosomal syndrome? When the changes in the phenotype of the organism occur due to changes in autosomes, then it is called autosomal syndromes. And when the changes occur due to the changes in sex chromosomes, that is X or Y chromosome, it is called sex chromosomal syndrome. Now before we start the study of syndrome, we need to know the perpetuation of the life through time. Here we can see what happens in the human. We have two type of cells. One sort of cell is called somatic cell, another is germ cell. Now when we deal with the genetical when we deal with the gametogenesis, we see that the meiosis cell division takes place in spermatogenesis and oogenesis. Now in spermatogenesis, the meiotic division takes place in testis, and in oogenesis meiotic division takes place in ovaries. When sperms and eggs are created, they are always haploid. That is, in somatic cells, the number of chromosomes is always diploid, but in gametes, the number of chromosome is haploid. Now, when these gametes are fused, one zygote is formed, and then many mitotic division takes place. Mitotic divisions take place, and then an embryo is formed. When this embryo is grown up, then what happens? We can see in any organism body, it is made up of so many cells. Now, when you see the center of the cell, you will find out there is a nucleus in eukaryotic cells. Here, the genetic material, that is DNA, is contained in a special structure that is called chromosome. Now, DNA have specific locations. These locations have the information, genetical information, to code a specific protein, and this protein, if is formed, forms a particular character. That specific location is called the gene. That is, we can say the gene is that segment of DNA which code for a particular protein in any organism that will define a particular character. Now, the DNA is always double helical. Now, Down syndrome, why it is called so? Actually, it is the name is given on its discoverer. The name of the discoverer was John Langdon Down. He was a British physician. He studied this disease and described so many characteristics of this syndrome in 1866. So, to give him the credit, this name was given. Now, what is the occurrence rate? Down syndrome occurs at a frequency of about 0.15% of all live births. That is, if 1000 children are produced, then out of 1000, 1.5 children have the tendency to suffer from this disease. Most affected individuals have an extra copy of chromosome number 21. This extra copy is caused due to the non-disjunction of the chromosome 21 in a parent who is chromosomally normal. That is, when this spermatogenesis or oogenesis process takes place, then meiotic division happens. Now, if during this division the chromosomes are not separated properly, normally, then it may create a problem. Now, which problem? That we can see with the help of one picture in the next slide. We know the chromosome number 21 is the smallest human chromosome, which contains 48 million base pairs. That is 1.5 to 2% of the total DNA in the cell. This diagram explains the process of non-disjunction. Here, at this place, we see the pair of the chromosome number 21. We know human contain 46 chromosomes. That is 23 pairs of the chromosomes. Now, the trisomy 21 is when there are 3 copies instead of 2 copies. In normal human being, only 2 copies of a particular chromosomes lie. So here in this diagram we can see that in parent there is only one pair, but when spermatogenesis or oogenesis takes place, then in above diagram we can see during first division the chromosomes do not separate normally, and these both of the copies go to one type of cell. This is the non-disjunction at first division. In another diagram we can see during the second division also this non-disjunction takes place. So here we can see in first diagram, at first division the non-disjunction took place, due to which second daughter cell got no chromosome, and then further these also got no chromosomes. While in the diagram which is mentioned below, here the parents had normal chromosome copy. In first division it was okay, and when second meiotic division took place, in one of the cell the non-disjunction took place, due to which one cell got no chromosome and one cell got 2 copies of the same chromosome. Now, what happens if this gamete gets the chance to be fused? That is, if it is a sperm or if it is ovum. If it gets the chance to be fertilized with its counterpart, then there is the chance for Down syndrome. Down syndrome is called as 21 trisomy, which is also called as Mongolism. Here in this syndrome, one extra copy of chromosome number 21, that is autosomal chromosome, can be seen, is found actually. The combined phenotype that make up Down syndrome include that is mental retardation. Here the IQ level is up to 20 to 50 range. A broad, flat face, eyes with an epicanthic fold, short stature, short hands with a crease across the middle, and a large wrinkled tongue. These characteristics will be studied with the help of a diagram in the next slide. Females may be fertile in this case. They may produce normal or trisomic progeny, but males do not reproduce. Mean life expectancy is about 17 years, and only 8% of the persons with Down syndrome survive past age 40. This is the diagram where we can see at in the karyotype here 3 copies of chromosome number 21 are present. So we can say if we see the karyotype, we'll find out 47 chromosomes instead of 46 chromosomes. This is an image which shows the human which is suffering from the Down syndrome. This is the male trisomy. This is karyotype, where all chromosomes are arrested in metaphase to study the genetical disorders. Here, the autosome 21 is with 3 copies, and the formula will be 2n plus 1, that is 47 chromosomes instead of 46 chromosome. And if it is male, then there will be one X chromosome and one Y chromosome. Here we can see the characteristics in detail. In the right, we can see the broad flat face, slanting eyes, epicanthic eye fold. Epicanthic eye fold means when upper eyelid gets fold near the inside, near the internal area of the eye, that is core. Here you can see at this place the skin fold is found. Here, very close to nose. So another characteristics are short and broad hands, small and arched palate, big wrinkled tongue, dental anomalies, congenital heart disease, there will be heart defect, by birth enlarged colon, colon will be larger in size, big toes widely spaced with other fingers, diminished muscle tone due to which they are unable to use their musculature for proper working, abnormal pelvis, umbilical hernia, that is swelling in the intestine, large intestine, intestinal blockage, uni or bilateral lateral absence of one rib, spatial skin ridge pattern, palm creases, that is we usually have two creases in our hand, but there will be only one crease in the person who are suffering from Down syndrome. Many loops on fingertips, flat back of head, and abnormal ears. These are some specific characters which can be observed in the person suffering from Down syndrome, that is trisomy 21. This is a graph showing maternal age and the production of offspring with Down syndrome. That is, if a woman conceive in early age, then there are lesser chances of Down syndrome, and if it goes to the later age, then there are chances for occurrence of the Down syndrome. The main reason is that in old age, the proper functioning of the proper functioning of the oogenesis will be disturbed, due to which the occurrence of Down syndrome is high. For this reason, fetal chromosome analysis is now recommended for the females who conceive baby at the later age of life. Here we can see the process is being displayed, how the karyotype study is done. In female Down syndrome, the autosomy will be the autosome 21, chromosome number 21 will be with 3 copies, and in the karyotype 2x chromosomes will be found. And here is the process. The process is the cells are taken out from the body, then they are cultured in vitro with the help of hypotonic Giemsa air drying technique. Then the slides are prepared for chromosome analysis. Here are the references that has been used for the preparation of this presentation. I'm thankful to you all for watching this video. Thank you. Have a good day. Have a nice time.